So again, glad that she decided to come out and join us. It's my great pleasure to introduce Dr. Marcella Kelly. She's an associate professor at Virginia Tech. She does a lot of work using non-invasive sampling techniques, using scat samples, using camera work. Marcella was actually my undergraduate advisor, and I didn't realize how much of an influence she would actually have on my life, on the type of research that I do. Also acknowledging that professors can be cool. She was the first professor that I was like, oh, okay, so she's like normal, like she tells jokes, and I see her with a beer, and like she's cool. Okay, great. So they're not all the same. So quite inspirational from that context as well. Hopefully you will enjoy her talk entitled Carnivore Coexistence in Forested Ecosystems.
Okay, thanks Naima for that. Is this working? Can you hear me with the mic and all that? Okay, great. Okay, so what I thought I would do today is, rather than just give one straight research talk, try to link a bunch of different studies together with a common theme. But first, to set up the background about why I'm interested in this carnivore coexistence and where it came from. It started really back when I was doing my PhD research on cheetahs. And this is actually a picture from Serengeti, and that person in the vehicle there is a current grad student of mine working on this same population. And so this was non-invasive photographic data that was collected by people actually taking photographs, so not remote cameras. So people taking photographs of cats, and then my PhD advisor giving me this data set of 10,000 photographs. So everyone's like, you worked on cheetahs. I'm like, there they are. In those boxes there. That was my life for about a year and a half. I did identifying individuals by their spot patterns. So I got all these cheetahs identified. I bought a Dalmatian somehow randomly in a mix of all that. And this was the work that I did at UC Davis when I was there. And just to set the stage, if you're unfamiliar with this ecosystem, cheetahs really suffer a tremendous amount of predation due to lions killing their cubs, and also lions steal their prey. So cheetahs will hunt a nice Thompson's gazelle, and then lions will come and take that from them. So kleptoparasitism and intraguild predation dominate in this, at least for this predator. So just looking and just doing a couple of brief slides on what I found from the PhD research and why I continued on this topic was that cheetahs before 1980 they did very well. Lots of cubs were produced. Post 1980, they had a hard time even getting one cub off to independence. So some of the PhD research showed that these litter sizes were declining post 1980, litter size at independence, lifetime reproductive success, reproductive rate. So all these reproductive measures were declining post 1980. And lots of that, most of that was due to predation. So most of the cubs were being killed by predators. The cheetah cubs only have about a 5% chance of survival. And most of that mortality is due to predation by lions. Hyenas do some, but the majority is lions. And so then what we also noticed in this population is this spike in this top graph here. This top line is female lions that are on the plains in Serengeti. So around 1980, female lions start to increase on the plains. So that got me into, and I was at that time was working on viability analysis. I was well entrenched in that kind of world of what could you do if you've got two predators in an ecosystem that are both endangered? Could one cause another one to go extinct? So back in this day of doing population viability analysis, we basically found that when you get up to 90, a little bit higher for lions, that cheetah extinction risk goes way up. So they really can't handle it. You've got this impact on their recruitment into the next generation, and once you get a certain threshold level of lions on the plains, cheetah population extinction risk is really high. And if you look at it in another way, here's just a picture of lion biomass on the x-axis, and then cheetah biomass corrected for prey, but that doesn't matter. The main point of this slide is just showing you that these are all national parks in Africa, and as lion biomass increases, you have a decline in cheetah biomass. So lions and cheetahs are, they can't coexist very well together, not in the same areas. So lions really could not be used as an umbrella species for cheetahs, because the predation rate is just so high. And lions have caused extinctions of other carnivores, so they've caused wild dogs to go extinct. So that's just a little bit of the background. So then I decided, finishing up my PhD here in the late 90s, early 2000s, and I want to look at this idea of trophic cascades where you have a top predator maybe removed from a system. In this example, this is the Crooks and Soulé kind of classic example of this trophic cascade. You take coyotes out of the population, and you get an increase in feral cats, and you get cats killing all the little birds. So not very good for the birds, and so I thought, well, we kind of had this thing going on in Serengeti as well. Let me just move to a different ecosystem, and I want to test this in a couple of other ecosystems. So that's kind of the background where I came from. So, and then you probably are all familiar with the whole idea of mesopredator release. So we've got, in the absence of a super predator, so my idea was maybe I can go try this in a different part of the world where we may have lower densities of super predators, like a jaguar, and then we can look at when jaguar densities change, do the mesopredators, so pumas or ocelots? Or is there some spatial avoidance? So we could look at, there could be spatial avoidance, or there could be just changes in density. So those are the kinds of things I'm trying to go for. But at the same time, we also have this other idea out there that predators are often used as an umbrella species. And that always kind of bothered me because I was like, well, which one is it? Is it trophic cascades and mesopredator release, or are these animals really umbrella species for all the other species in the environment? And depending on who I was writing a grant proposal to, I would just use one or the other, right? So umbrella species to the NGOs, mesopredator release for NSF, even though these are like totally competing ideas, and I think they can't both be happening. That led me to look it up back in the day, and apparently jaguars are an umbrella species. There's not really actually been really much study on this, but they're defined by Wikipedia as an umbrella species. So I was like, all right, let's go see if this is really the case. So now with that background, I'm going to go into 3 case studies that are all kind of with similar techniques looking at carnivore guilds. And so the first one will be the jaguar puma ocelot one and where we are with that. The second one is the Sumatran tiger, clouded leopard, and 4 other cat species that I'll talk about. And then we'll end with just a little bit at the end about Madagascar and the Malagasy carnivore community. And so all of these study sites are kind of linked by these overarching ideas, and we're using all the very similar techniques in these study sites. So data is really lacking on carnivore coexistence. It's really tough to study. Once you get outside of Serengeti. Serengeti is one of the few places in the world where you can actually watch carnivores, see them out there. But in places like the tropical rainforest, it's really hard to see these cats. And they're just very, very difficult to study, really elusive, wide-ranging, nocturnal. So all these things that make them super tough to study. So as part of a side project that I was doing as part of my PhD, actually, originally it was a completely failed PhD project. We can talk about that later. So I quit a project that didn't work at all and then went back to the cheetah project. But I worked in this country in Belize and I really liked it, and I thought, we can go back and do that now. I've recovered from the trauma of failing my first PhD attempt, so now I can go back in and tackle this question of mesopredator release or umbrella species, et cetera. Okay, so small country, and here are the techniques that at the time were not really used. So this was some of the new up-and-coming things in the late 90s, early 2000s was the advent or the sort of widespread use of remote cameras. So those were becoming a thing. I'm also going to talk about the idea that you can also use sign surveys, so tracks, scrapes, scats, but also genetic methods as well. So I'll talk about these methods, including scat dog methods as well. So I was armed basically with this idea that I was really interested in, and then this paper by Ulas Karanth and Jim Nichols. Jim Nichols was one of my mentors, and so he and Ulas published this paper saying you can actually do this thing with remote cameras. So you can do mark recapture, you can set your cameras out there across the landscape. Tigers are individually identifiable by their stripe patterns, so we can, I was thinking I can do this. Somebody else did it first, I'm just going to do this now on jaguars, ocelots, and things like that. So here I am armed with my first WCS grant and my, I think it was 14 cameras. I'm going to tackle the world. So here they were, the old film cameras, if anybody remembers what film is. There might not be too many, but what a pain in the butt they were. Lots of troubleshooting with film cameras, a big learning curve working in tropical rainforest. I had been there, that's a very different thing from trying to work in them. Very difficult, muddy conditions constantly. I'm very allergic to poison wood, it would appear. I break out in terrible hives and have to go to the hospital. So that's something else I learned. I got many more trucks than this stuck in the mud. That's a rental car there. Yep, sometimes it can be a little bit dangerous. You might have to go with Army guys to keep you safe. You might start a little war with the Guatemalans because they're stealing your cameras. So this was the Cold War that we refer to in Belize, of trying to bolt our cameras into these concrete pods to make sure they didn't get stolen. And eventually I lost the Cold War and I had to leave this study site, sadly. And then if anybody hasn't checked out Twitter and looked at Fieldwork Fails, this is one of the postings from Fieldwork Fails where one of my grad students quit after this. My only grad student I've ever had quit. So it was just not his cup of tea. So anyway, and that was also a rental car too. So this was a large learning curve. It would have been nice to have someone to teach me, and I was also starting professor at Virginia Tech, so this was like a lot going on. But eventually, we got it all figured out, except there was one very important graduate student who helped me do it. So one of my first graduate students helped me get all those cameras into the forest when there were no trails. So we would cut trails and do all this kind of stuff, getting the cameras there. Okay, so here is just a brief style of how you set up your study design for a lot of these remote camera projects, and this is going to be pretty similar for all of these projects. So I won't repeat this, but if you're not familiar, this has kind of now become standard methodology for camera trapping. And in this case, the minimum that we would start with would be about 20 camera stations. Now we're up to 100 in certain study sites. In my case, we're using 2 cameras per station because we want to get both sides of the animal because they have unique spot patterns on both sides. And the spacing is going to depend on what your target animal is. So we did 1.5 when we were looking for ocelots and ocelot density, and about 3, or it's really about 2.5 for jaguars. The cameras have got to be out there long enough that you can get enough data back that you can do mark recapture. So you need to get a lot of captures and recaptures. But you also have to satisfy the closure assumption, so the population's got to remain closed and all that kind of stuff. So it takes about 2 to 3 months to balance that trade-off of closed population but needing enough samples that you can do mark recapture. And then you can run, and I'm skipping like a lot of the nitty-gritty, like all the nitty-gritty details of mark recapture, but if you want to talk to me about that, there's so many new techniques coming out with spatially explicit models and all kinds of stuff that we're working on in my lab that we can talk about that later. So when it's kind of all said and done, in this case, we've got 7 study sites across Belize, and some of them are very close together, but if they are close together, they're separate because there's a different management style, and there might be logging in one reserve and not in another. And so we've got grids across this whole country just about. And then when you buffer those grids with, there's all kinds of buffering techniques, but suffice it to say, we're surveying 200 to 500 square kilometers of these forested habitats. Okay. Oh, yeah, you always get some funny pictures back. We've got to show a funny picture of a tapir peeing on our camera because that happens actually fairly often. So we get, and I'm not even going to really talk about non-target species. We get lots of non-target species as well as the cats that we're after. So here are, in this particular study site, here are the 4 other cats. So we've got ocelots up here, little margays, pumas, jaguarundis. Again, the spotted cats are individually identifiable, so you can tell them apart. It's much harder to do individual ID with the non-spotted cats, but I'll talk a little bit about how we're trying to get estimates for these guys in just a minute. And then you get all your jaguar photographs back, and then we spend a lot of time, or I would say actually the undergrads in the lab spend a lot of time matching the cats up and helping out with keeping the long-term database running. Okay, so, and here's just a kind of a little anecdotal slide I wanted to put in because this is just an example of a cat that is actually traversing among multiple study sites, so they can move between the study sites, which sets up some interesting analysis problems. But in this case, this guy is moving from these logged areas in yellow. All of these are logged, the orange, sustainable logged in red, and to this area up here, which is actually unlogged. So this particular animal is moving very large distances, and the reason I put this up here is to show kind of his happened to the animation. Well, there was an animation in there. Oh, there it is. Okay. So there is a very long distance movement that he made. And this is kind of important because previous to this time, the only estimate of home range size for jaguars estimated home range size at 40 square kilometers, which all of us knew was like really way too small. And so here we've got an animal that just has moved a straight line distance of almost that distance. So we know that they have much bigger home ranges than previously recorded. It's just that the style back in the day was just telemetry collars, so it was probably not very accurate estimates. Okay, so that's just a little anecdotal, interesting information. And skipping a lot of the mark recapture, the main reason that I went right to this slide is to show you that there is extreme amount of variability in jaguar densities across these 7 study sites. So in the low here, Fireburn up in the north, from 2.3 animals or 2.4 per 100 square kilometers to the high, which is still the highest recorded anywhere in the world, is 10.24 jaguars per 100 square kilometer. So that's a really high density. So believe it or not, for those of you who don't know cat research or carnivore research, that's pretty high density. Okay, so that gives me a little bit of a range now to look at, do the ocelot densities vary? So if we're looking at ocelot and puma densities, are they going to be tracking or are they going to be opposite if mesopredator release is happening? So just to kind of go into some of those results, we haven't finished yet, but I'll give you some of our preliminary assessments of what's going on. And in this case, this is just a trapping rate, and what we did was, well, let's just do a first crack at this. Let's look at areas where ocelots were present and ocelots were absent. And it turns out at the present site, we have much higher trap success of jaguars. So again, this is just presence, absence data, but it's interesting that it's the opposite of what we expected. So we were expecting ocelots would avoid jaguars, or areas where there were a lot of jaguars, they would have fewer ocelots, but instead we're getting the opposite. There weren't really any relationships at all between ocelots and pumas, or pumas and jaguars. And I'm going to talk a little bit about occupancy analysis. If you're not familiar with that, this is really just using presence-absence data again to look at distribution across the landscape while you're accounting for imperfect detection. So in this case, the green is one of our logged sites, and the orange is one of the unlogged sites. And the main take-home message for this slide is that there's not really any difference among the carnivores. So areas that have high occupancy, like these unlogged site, for pumas at almost 100%, it's the same for ocelots, it's the same for jaguars. So they all do better in that, or they all at least distributed more evenly across that unlogged site. And then in this case, in the logged site, they all have a little bit lower occupancy, but it's the same across the board for all the species. So again, opposite to what we thought. We thought that there would be some spatial partitioning. If an area had a lot of jaguars, they would have lower occupancy for ocelots and pumas. So we're not getting anything from that in terms of the mesopredator release hypothesis. So for some of the density estimates, now we haven't been able to do pumas because they're really hard, and I'll talk about that in a minute, but this is just 3 of the study sites that we finished. And what we're showing here is blue are ocelot densities, and the maroon color is jaguar densities. And basically, if this site is bad for the site here in the Mountain Pine Ridge, this habitat is just bad for both. So there's very low densities of jaguars, very low densities of ocelots. And then this site here has the highest density of ocelots, the highest density of jaguars. And this site here has intermediate levels of both cats. Granted, there's some overlap in these confidence intervals, but the take-home message is that we're just not seeing it, at least in the jaguar ocelot. There is a cat in the middle, so there is, I know we're missing a trophic level potentially. But let me just show you some of our long-term data that one of my students has been really cranking out these ocelot density estimates. And again, the top two panels are from the logged site. The bottom two panels are from the unlogged site. And what we're showing here are just ocelot numbers in red. And these are density estimates per 100 square kilometers. So ocelots in red, jaguars in blue, and we really are seeing almost a tracking of these population densities. So in years that jaguars seem to do well, so do ocelots, or there's not really a relationship. And I ran these as regressions, ocelots versus jaguars, and they're all positive, so there's no negative associations. They're not significant, but so we're, again, seeing what we didn't expect was this sort of tracking of the cats. So when one cat does well, so does the other one. Okay, so skipping a bunch of AIC modeling, and I'll just talk about when you incorporate some of these habitat variables into the models. So it could just be that the cats are all choosing the same habitat. So that's what's driving these relationships. And we did find that within that pine forest habitat where the cats, none of them did very well, there was some habitat variables that popped up, like road width was actually positive. So animals all used wider roads, which was strange. We were not expecting that. But then again, these are logging roads that are old and not used very much. Human use did have a little bit of negative impact, but when we added the trapping rates of the other cats, we got better models. So it's like there's also something going on with that we're not picking up on some other habitat variables. And the same kind of in the broadleaf sites. Positive relationships between pumas and jaguars, mediated by positive with road cover, with canopy cover and roads. And ocelots had positive associations, again, with pumas and jaguars with some road influences. So basically, we're seeing all positive associations with these cats, even when you add the habitat variables. So it's really, even when you add the habitat variables, the models with the other species being trapped are still better. So there is something interesting going on that is opposite to what we expected. I just kind of want to wrap up a little bit of this association with this study site and talk also about where we're going with this. We're almost done with all of the ocelot work, at least up to 2015, and we've now got these data sets with lots of data where we're using spatially explicit mark recapture models and trying to advance some of that stuff. And then what we're not as far along on are the puma mark recite models, so you can identify some of your pumas in your population. And here's an example up here of a really messed up puma. So this guy's bumps all over his body are bot flies. So those are parasites that are going to pop out eventually. So we can identify parts of our population, a proportion of that population, and then with that data we can use that information for the unmarked animals that we can't tell apart. And then we can get some pretty decent estimates of puma density. So we're in the middle of that, and when we add that middle predator, I hope that everything becomes more clear. But I'm suspecting not. I'm suspecting that all the pumas are going to track the jaguars and ocelots at this point. Okay. Another part that I wanted to talk about too, because I think this is interesting and a lot of people are unaware of the sort of scat collection methods that you can use now on these large cats. And in our case, we decided to go with genetic monitoring and picking up scat samples. And we chose to use 2 scat detector dogs. So this is Billy and Bruiser here, and these guys were trained on all 5 cat species. So they're trained to find all those poops, and then we take a sample from them and send it back to a lab. Both of these dogs were, a lot of people ask, where do these scat dogs come from? They usually come from some company. We went with a company called Pack Leader, and in this case, Billy was he was a former bomb dog, so he got retrained on our project. He had worked in Iraq for a couple of years, and they said he had PTSD and he couldn't do Iraq anymore, so he was out of there. Bruiser's funny because he was probably our best scat dog, and he was really good in drug detection. He was going to be used as a drug detector dog, but they couldn't use him because he flunked out because he would go into a house and destroy everything, jump on the tables, run over the whole house, and break the lamps and all that kind of stuff, so they couldn't control him, so they said, outside dog only. So he got trained for us on scat detection down in Belize. Okay, so interesting side note. There's a lot that goes into training a team for a scat dog team. So this is my student Claudia here. She got trained as a handler, and she worked with this dog for a couple of years. And at this time, there was nobody to tell us how do we go survey for doing mark recapture with a scat dog. There was no person to call or ask, and so we just came up with a bunch of different ideas. And in this case, we just put these red squares over our camera trapping grid, which were about 4 square kilometers, and then we would search through these grid cells, picking up all the poop, and we would consider once we finished searching those grid cells, that would be 1 capture occasion. Then we'd go back in and we'd start all over. Survey again, 2nd capture occasion. So it would take us about 10 days to get through the study site, and at the same time we've got the cameras going. So this is how we built our kind of mark recapture models. Billy was very polite. He would point. I mean, he would actually sit and say, "There's the scat. There it is." So, and then they do this all day just so that they can get their ball. So that's the reward. It's nothing more than that. So they definitely need to be these ball-driven, kind of crazy dogs that are really into an object. Okay. So with the scat surveys, what does that look like? In this case, we've got really the main thing to pay attention to is just that we got over 1,000 scat samples. Claudia surveyed. She walked about 1,500 miles if you count all the backtracking. Here it's in kilometers, but and she picked up about an average of 2 scats per kilometer, or 4 or 5 per day. So she was getting a lot of scat samples, and we estimate that probably about 75% of those would have been missed if we didn't have the scat dog. So very hard to see by ourselves. The dog's nose is just so good, and they just hit on little things that to us might look like a pebble or a rock, but the dog hits on it, and then we would collect it, and it would amplify in some cases. Amplification success is a little bit low, but we were able to get a bunch of pretty cool results. So here are some of the results from that. We ended up, these are all the study sites here, and then we ended up with 74 jaguars. So that was we caught 74 different individual jaguars 307 times. The technique was pretty good for pumas too. We got 56 individual pumas in 161 captures. Was not as good for ocelots, so we did not get as many ocelots. We got way more ocelots with the camera traps than we did with the scat dog. And then here's just some of our amplification success and things like that. Okay. And just to do a couple of really interesting comparisons here, what was, I thought, pretty interesting was we thought that we were going to get more females with the genetics because the dog can go off trail and all over the place, but we did not. So in this case, these are just 3 different study sites, and the blue is male jaguars, the pink is female, and sometimes in camera trap data you can't tell if it's a male or female. So with genetics, we were really missing those females. So we got them, not as many as we did for males with camera trapping, well, at least not at 2 sites, but at 1 site we got more females than we did males. But the genetics, we just keep missing them. And so that was a big, interesting learning lesson, but it also is possible that the female DNA was not as good quality because we're still kind of analyzing and trying to figure out if those ones that didn't amplify those could have been females, or maybe those were just the degraded ones that were off in the bushes rather than the males really, we think, mark very distinctly in the middle of a trail. So anyway, some interesting results. Of course, when you convert those to densities, what it means here is that overall we got more animals with the camera trapping than we did with the genetics, which was not what we thought. So our densities were higher with cameras than genetics. So in a way, Claudia and I would fight about this all the time, who is going to be better, you know? And so I'm not going to give up on camera traps. I think they're still actually better than genetics, potentially for doing density of these kind of animals that have natural markings. But the thing that you can't do with that you can't do with cameras that you can with genetics is look at this landscape connectivity, which is really cool. And again, this is a little bit out of my area of expertise, but Claudia really took this on as her baby, and she became kind of a geneticist through the process. And so we were going to look at connectivity across this landscape using those poop samples that we picked up off the ground. We do know that ocelots get hit by cars, so we've seen animals get hit by cars. We have no idea why this jaguar lost his tail, but he had a tail and then he lost it. Maybe there was some anthropogenic disturbance, maybe, I'm not really sure. So there is fragmentation, there is human impacts. So do we need corridors for these large cats? And so here's just some of the FST values. If you're not familiar with genetic results, which I wasn't when I started this project, the higher the number, the worse it is. So that means the populations are subdivided. The lower the number, the more inbreeding, the more movement there is, and the animals are breeding together. And so we had, interestingly, really low FST values. So most of them were below 0.05, except for a couple of cases where we had moderate. But in general, the samples that, and these are the number of individual jaguars that we got across this landscape, are all basically doing pretty well, and most likely it's one single population. But this is really interesting too. This is the graph that's depicting parent offspring or full siblings moving from one reserve to another. So in other words, these black lines indicate that we got a parent in this reserve, and then we got its offspring down here, or vice versa. We can't tell which is the parent and which is the offspring. And the same for full siblings. So we end up getting, the red lines are siblings, and how many times we got that. So we had a lot of these instances of, we caught one cat here and its brother up in another reserve. So one of them moved, and so this is really amazing that we got this much movement of the young across this habitat. I'm going to skip the assignment test because that takes a while to explain, and just say that basically we think that for now it looks like panmixia for jaguars. Pumas are doing pretty well as well. They seem to have a little bit more subdivision when we do the FST values. They're a little bit higher. Ocelots we weren't really able to do much with. We only had 30 genetic samples, and we couldn't really spread those across the landscape. So we don't really know for ocelots, but jaguars look pretty well, pretty good. Pumas look pretty good genetically. Maybe there's some hints of potential issues that I don't have time to talk about. Okay, so that was sort of an overview of some of the just genetic techniques that we're also employing that didn't really directly address this question of whether or not we've got mesopredator release going on, but I thought it's really interesting to show a little bit of that work, and I think we can tie it in later to see if there are any genetic breaks for some species and not others. So what I'll do now is switch gears on you and move into the two shorter pieces of this talk where we use this broad scale approach with camera trapping, not genetics, just to look at this occupancy stuff across the landscape, and mostly occupancy. Okay, so here's the Sumatran tiger example, and this is a very large landscape here, this is our grid that I'll explain in a minute in central Sumatra in Riau province. And what we did in this case was we had this occupancy grid. So these grids are 17 by 17 kilometer grid cells, and they're sort of stratified random sampling. So we've got some yellow ones that are in the flatlands and plantations, and then green ones in the forest. And we surveyed these in a style so that we could do occupancy modeling. So we did repeated surveys. Each one of these crosses represents transects that we did, and so we did 40 of these transects, and then we used those as our spatial replicates. So we didn't have time to go to these study sites repeatedly through time. We only had time to go there once, so we did spatial replicates. So we would do 40 different replicates of these transects looking for all kinds of tiger sign and other species sign. And then we would collect a whole bunch of habitat variables. So I'm not going to go through the habitat models, but just suffice it to say that tigers, we did get a lot of tigers detected in, they definitely prefer covered sites with pretty thick understory. They preferred forest. However, they can use plantations as long as they're older. So these are, I'm just skipping to some of the results of the first level of occupancy before I go into the species interaction stuff. So they can use acacias sometimes. And they do like sites that are closer to water within those plantations. So those are some of just the little bit about tiger ecology. So, and then you can use these occupancy models to predict tiger occurrence across this landscape. So the darker areas are where we would expect tigers to occur. The lighter areas, most likely they're not really occupying that area. So the neat thing about using this type of occupancy modeling is you can extrapolate your results across a much larger site. So we only surveyed 47 blocks, but we could extrapolate across all 180 using the same GIS variables that we put into our models. Okay, so that's just a little interesting side note. But to get back at this question that I really was interested in, these felid interactions, here are our players in this system. And again, this is another example of just wild cats. So we've got tigers, clouded leopards, golden cats, leopard cats, marbled cat, and flat-headed cat in this analysis. And what I wanted to do is take a different approach. We've gone a little bit farther with this analysis and say, you know, where do these carnivores overlap spatially? And does detection of one influence detection of another? And so if you're not familiar with these two species occupancy models, basically we're looking at both detection and occupancy of these two animals. And when you account for the detection of the other species, does that change your occupancy results for the species of concern, or the less dominant one? And so how they're structured is you need to kind of decide who do you think is the dominant species, and then you can look at the lesser dominant ones. And one of the outputs for occupancy co-occurrence models is the species interaction factor. The main thing is that if that species interaction factor is greater than 1, these animals co-occur together more often than by chance. If the species interaction factor is less than 1, then there's some kind of repulsion. They're not occurring together, or they're occurring together less than random. And so this is the approach that we took. And so now what I'm doing is showing you that occupancy results for these co-occurrence models with the cats ordered from Sumatran tiger down to what we consider the most dominant cat down to the least dominant. So tigers first, clouded leopards, golden cat, leopard cat, and marble cat. And these are basically almost based just on their size alone. I don't have time to show all the little details of the AIC models, but what I've highlighted in green here is that we found no negative interactions. So no SIF values were ever less than 1, so they're all greater than 1. Most of them had these very large error bars, though, so the error bars overlapped 1, so we can't conclusively say anything really about those two species. They're probably just independent of each other. But interestingly, the leopard cat loved everybody. So the leopard cat was, again, a positive association, not what we were expecting at all, between leopard cats and every other cat. So leopard cat and marbled cat was positive, leopard cat, golden cat, clouded leopard, Sumatran tiger. So all these green guys was really the only thing that came out of it. Again, a positive association, totally unexpected again. And we were like, wait a minute, there really aren't these negative associations, at least in space. So what about time? Maybe we can look at temporal separation of these cats. And so we did this with kernel density estimates on circular data, which is time of day, essentially. And the delta in this case is the coefficient of overlap. And so what you try to do is you try to take your time of day that your species is active. So in this case, here's noon up to 24-hour clock. So in this case, this animal's not very active. This is a very nocturnal animal, not active during the daytime very much. So then you can take your other species that you're interested in and try to overlap these two on top of each other and then calculate that delta, which represents the temporal overlap. So the higher the delta, it goes from 0 to 1. If it's 1, it's 100% overlap. Okay, so when we do that, I'm going to do the same kind of slide here where we've got the cats ordered from largest to smallest. And in this case, it doesn't matter that much that the dark line is tiger and the dashed line is clouded leopard. The main thing I wanted to do was see is there any evidence of this temporal separation. And so I decided to just go along these diagonals. And when you go along these diagonals, this represents cats that are the closest in size to each other. So on that outer edge, Sumatran tiger and clouded leopard are closest in size. Clouded leopard and golden cat are closest in size. So this outer edge here are more similar in body size. So their overlap was 0.62 in time. So their temporal overlap was 0.62. What about the next layer down? Much higher overlap. So there does seem to be potentially something going on here where these animals, when they are more closely linked in body size, they have less temporal overlap. As you start to see some separation, there's more and more overlap as the body size differences increase. And the same if you continue down the chain. So the lowest overlap came from these similar sized cats. So I thought that was at least something interesting. We're finding there's not really any spatial component to this overlap, but there is in the temporal overlap. So maybe that's what they're doing. So for Sumatran felids, some evidence that they actually co-occur together, not really avoidance, some evidence of temporal segregation. There are some areas that harbor all the species. There are some areas like peat swamps that don't have very many species. And that potentially this diversity in this cat species could be promoted by just having sort of different mosaics of habitat. Okay, so that was just the quick and dirty on how far we got with felid coexistence, and I'll do this similar thing one more time for another forested ecosystem and a different suite of carnivores in Madagascar. And so these guys are, well, first, the study site that we've been working at since about 2008 is up here in northeastern Madagascar, Makira National Park, Maswala National Park. Again, camera trap data, similar analyses. And we had multiple study sites. In this case, again, we had 7 study sites, and 2 of them were surveyed multiple times. So I'll show you some of the longer term data as well. And again, carnivore occupancy analysis primarily. And Madagascar's got a lot of forest loss, and it's a tough place to work, just suffice it to say. So lots of human encroachment. Probably the most difficult thing is a lot of bushmeat hunting, a lot of poaching. And this one is the one that I'm going to focus on a little bit more today, because we do have exotic carnivores that are mainly in the form of feral cats and feral dogs that are moving into these ecosystems. Okay, so let's look at the players here in the carnivore community: the Fusa, civet, Falanuk, ring-tailed Bontsira, broad-striped Bontsira, all these little cute little animals that nobody knows anything about. So we tried to tackle these with our camera trapping, and we've also got now these other introduced species, so dogs, cats, and Indian civets. So we've got actually a very diverse carnivore community here. And so because I've talked a little bit about occupancy modeling already, you kind of know the deal. We did these 2 species occupancy models. And rather than present all of them in one big diagram, because there would be many more than the Sumatran diagram, I'm just going to show you the results that we found that were really interesting for this study. And so this one is domestic dog or feral dog, and the Falanuk. And so what this graph is showing you is that you've got patchiness. So this x-axis means that as the numbers get higher and higher, you've got a more patchy, fragmented forest. So lots of little tiny patches versus one big forested ecosystem. So that's how we decided to, one of the ways we decided to look at fragmentation. So how patchy is that landscape in that camera trapping grid? And we combined all the sites together. And so this is just showing you that this co-occurrence modeling, what we've got here is occupancy of this native carnivore is much, much lower when you've got dogs in that ecosystem. So the occupancy is really very low, from almost gone to only up to about 40%. When you don't have dogs in an ecosystem, or when they're not present across camera traps, you've got much higher occupancy, so 30% up to 80%. So it's really interesting that this is, again, across all the grids, the dogs, and this also bears out in the same analysis of that co-occurrence thing, that species interaction factor. So that's what this graph is showing. Again, if you're at 1 means independence, greater than 1 means co-occurrence, and less than 1 means avoidance of each other. And so we've got very strong negative interactions. So I finally got a negative interaction, but it wasn't the way I thought it was going to be. It's actually with an introduced species and a native species. So that was a bit depressing. let me look a little bit about, since we have some long-term data, let me just show you a little bit of the results from that. This is repeated surveys now, multi-season occupancy, so when we do occupancy through time. And what do we find? So I'm going to actually skip how you do that. If you want questions about robust design, occupancy modeling, talk to me later. So what we did here was hopefully not just freak my computer out. Okay, there we go. So this now is occupancy through time. So we've got years on the x-axis, and we've just got occupancy for each different species across these years. And the sad story of Madagascar is this. Here are 6 native carnivores. Only 2 of them have not had dramatic declines in the past since 2008. So the big one, the Fusa, seems to be doing okay. The next biggest one, the Fusa fusana, Malagasy civet, seems to be doing okay. Dramatic declines in all the rest of these species down to close to 0. So some of these species are really being hammered, and we thought initially it might be the dogs, but the dog occupancy has stayed about the same. They tend to go in the forest with the humans. What has dramatically changed is the cat community, the feral cats in the ecosystems. From 0 occupancy in 2008, we had no photographs of cats, up to over 60% or 65%, 70% in some cases occupancy in 2015. So again, it's correlational. We don't really know, if cats and dogs are causing this, but it is very troubling to see these kinds of results with your native populations tanking while, in particular, these cat species are increasing. And the feral cats here are about the same size, actually maybe even a little bigger than this second species here in Madagascar. So all these other carnivores are smaller than feral cats. Okay, so that's the sad Madagascar story. And then where I'm going to go, I've got a couple more slides that are just going to show you how can maybe we can combine space and time. Ooh, like the space-time continuum. Can we do that to figure out this, to look at this, this idea of carnivore coexistence? It's probably both happening. There could be space, there could be time. When we look at occupancy modeling over space, we're just looking at any time that day that the animal was captured, or sometimes it's even a week time period. So maybe we need to be more fine scaling this thing and look at space and time together. Where was the animal caught and at what time relative to the other animal? So here's where we're starting to go with the spatiotemporal value, which is something we've been working on in the lab. That's maybe we can multiply the species interaction factor that we got from our temporal models by our kernel density estimate from our, our spatial models, by our kernel density estimates from the temporal models, multiply those together to figure out across this landscape where do these species have a high possibility of interacting. And so this is our first attempt at trying to get at this idea. And again, this is the 3 axes now. Fragmentation on this z-axis, hour of the day, so time on this x-axis, and then on the y, we've got our spatiotemporal value of multiplying our deltas by our species interaction factors. And when we do that, we get something really cool that we don't exactly know how to interpret yet. But we're working on this with a bunch of different species, and what we think is that if one species has not nailed another species and caused it to go extinct yet, we've got, in this case, high probability of this introduced species interacting with this native species, particularly at this time of day, across very fragmented habitats. So at fragmented habitats, this is going to be dangerous for these species interacting in the sort of late afternoons. And we have other ones that we've done with domestic dogs, which has shown that if you can keep dogs out of the forest at certain times of day, this potential for interaction we might be able to reduce it. So there are certain times and certain places that native animals are that if there's some way we can keep some of these exotics out, it could be of use for conservation. So that's where we're going on that. And so to kind of wrap everything up, jaguar conclusions, the jaguars do seem to be doing very well. They do seem to be an umbrella species for at least ocelots, most likely pumas. We're not finding evidence of mesopredator release. So maybe they could be a good umbrella species. For Sumatran tigers, same thing. The co-occurrence models revealed no evidence at all of this sort of spatial separation. So they also could be good umbrella species. And then interestingly, the fossa might not be a good umbrella species, but for a very different reason than the previous studies. And that is just because we've got introduced species that are coming into this ecosystem and messing things up. And so the negative interactions there are really caused by a non-native species that has recently been into these habitats. So, and we can't account for that just by using the fossa. So the fossa is not really a good umbrella species in this case due to other reasons, not mesopredator release though. So anyway, so that is kind of trying to wrap all of that up. And I think that cross-ecosystem conclusions, I would just say that this high intraguild predation does really affect cheetahs and lions, or does affect cheetahs, but is that generalizable to tropical forest ecosystems? Maybe not. Maybe that's really a thing of savannas where a lion can see a cheetah, knows she has cubs, goes over and kills them. You're not going to be seeing that in these forested ecosystems, so they may function in very different ways. And so we've got little evidence of intraguild predation, little evidence of spatial partitioning, some temporal partitioning. And then I guess the last couple of ideas is just that we can really learn a lot from this non-invasive sampling, and I didn't know how far we could take it, and I think there's still even more that we can do with these non-invasive methods, and we just need to be more and more creative. So it's a great time to be studying using these non-invasive techniques, because we've got a lot of capture mark recapture techniques that are really advancing very quickly, lots of ways to do sample collection. We're now working also on integrated models where you take the data from your camera traps and you integrate that with your genetic data to try to get even better estimates of abundance and density. And then we can also combine techniques. So I'm not just a non-invasive person. We just finished a project in Virginia where we're combining GPS collar data with genetic sampling. And so that one, I don't have time to talk about it, but that's actually a project here that a PhD student of mine just finished. So if you're interested in how to do that, I'd be happy to talk about that later as well. And so with that, I will just say thanks a lot for your attention. I crammed a lot in there, but if there's something that you want to talk to me about later, please feel free to come up and ask or ask now. So thank you very much.
Marcella, I'll let you field your questions.
Sure. Yep. Yep.
So, wow, this is really impressive. I mean, the amount of work you put into getting your data makes me feel really bad. I always thought I put a lot of work into getting data, but this is incredible.
It's somewhat painful, yes.
So I had two questions, one short one and one long one. So the first one is, what's the effect of habitat degradation, logging, etc., on the predators in the leaves? So that's sort of like I think that's sort of like very practical, the next question. The second one is, so we talk about intraguild predation and about how co-occurrence may tell you something about how species interact or whether they. And what I don't understand, I mean, isn't it possible that co-occurrence is influenced by a completely different set of habitat characteristics, the composition of the prey community? I mean, could these be like much more important than direct interactions?
So your first question on the logging, the quick answer is there's not much effect of the logging not on the predator densities. They're the same, very similar in the logged and unlogged site. That said, they are sustainably logged, so there is a single tree extraction. It's it's not clear cuts. So and they've had that in place for a long time. Interestingly, though, some species do better in the logged area, like white-tailed deer. So so that's kind of cool. And then the second question is that, yeah, we need to include more habitat data. We do have information on diet, but it's just locked in a freezer of poo that needs to be looked at and needs to be separated out to see how are they partitioning dietary, what is their dietary partitioning. They do eat really similar things, especially jaguars and pumas. That's been found in a lot of other studies. And ocelots, I'm sure, are eating much smaller prey. But that's a great question, and I think the only answer I could give you is on the most extensive modeling we've done with the habitat data comes from Belize, and our models were still improved when we put in the presence of a predator, the other predator, so that actually bumped it up. So there's something we're missing. It's not just habitat, because I don't think it's that, oh, there's a jaguar there, I'm going to go there. There might be, I think there's something else we're missing in this system, and maybe it's prey density. So that, we haven't tackled prey density at all because it's hard enough tackling predator density, and prey most of them are not marked. So there's really hard to do density estimates. But yeah, we just need another small army of people to work on the prey. So great question though. Yeah.
Have you taken any look at, say, just general food measures like EEB to look at the
Oh, yes.
fluctuation and productivity that might correlate with
Yeah, not in Belize. We have not done that, but we have done it in Nepal. We have a very similar project. I didn't get to talk about that one, but it didn't really come out as that important. I think with a lot of these tropical areas, there's not a lot of difference in DVI. It's really very, I think that's one thing. If I was to restart this study all over again, I might pick an area that had more variable habitat types. I think part of our problem is that the habitat is, even though their densities are very different, there's nothing structurally very different about the habitats. The trees are all very similar heights. There's a lot of canopy cover. I mean, there are some areas that have a little bit of wetland here, but in general, the tropical broadleaf forest is pretty similar, with the exception of that one pine forest habitat, which is tropical pine forest, and has very different animal densities. So, but yeah, I'm all ears about any other potential measures that we could use. Yeah.
what about this effect of the roads?
Yeah.
Be introducing it for them?
The road one makes me actually more worried than I ever talk about, because we do not survey randomly. We survey on those roads because you get really good higher capture success. And so